Hi everyone, welcome and thank you for joining today's webinar titled Exposure to Simple and Complex Carbohydrates in Human Milk and the Developing Infant Brain. My name is Haneen Abu Layla and I am the country coordinator for the Jordan Nutrition Innovation Lab. I am really thrilled to open our first webinar for this year and happy to see more people joining and familiar names. As more attendees are joining, I will be going over some of the logistical items. Next, please. I would like to direct all attendees to a few functions on this Zoom call. At the bottom of your screen, you should see a chat icon and a Q&A icon. Please use the chat feature to engage in relevant conversations with the other attendees. Next, please. If you have a question for our speaker, please use the Q&A feature. We have allocated the final 15 minutes of this webinar for Q&A, at which point our speaker will respond to any questions from the audience. This webinar is being recorded and will be made available on our website following the event. We will repeat these technical housekeeping items in the chat throughout the webinar as people may be joining in later times. Next, please. Okay, before I introduce our moderator for today, I would like to give a brief introduction to the Jordan Nutrition Innovation Lab. The Jordan Nutrition Innovation Lab is a research and capacity building program that is funded by USAID. By USAID Jordan, under Feed the Future initiative. We pursue research and capacity building activities to support the health and nutrition of women of reproductive age, pregnant lactating women, and infants and young children in Jordan. Some of our key objectives are implementing a rigorous maternal and child nutrition research agenda, including conducting analysis of existing nationally representative datasets and conducting comprehensive evaluations on programs such as USAID's Community Health and Nutrition Program. Moreover, we build individual and institutional capacity through the award of fellowships, scientific symposia, workshops, and webinars. As part of our capacity building agenda, we recently released the call for application for our third round of fellowship program. Through this program, a 12-month fellowship will be awarded to 4 emerging Jordanian scholars in the maternal, infant, and young children nutrition field. With the general support, the fellows will conduct their studies and complete publication-ready research in this field. In addition, we conduct these webinars on a regular basis to conduct outreach to a wider stakeholder group in and outside Jordan. This webinar is moderated by Dr. Len Osman. I would like now to introduce Dr. Len. Dr. Len is the Saqar bin Muhammad Al-Qasimi Professor in International Nutrition, as well as a professor at the Friedman School of Nutrition, Science and Policy at Tufts University. She has been an active investigator in USAID-funded projects in Malawi, Uganda, Nepal, and most recently in Jordan as part of the Jordan Nutrition Innovation Lab. She has been active in experimental studies with humans on lipoprotein responses to several vegetable oils, trans fats, and soy protein, and the glycemic response as modified by various meal components. Most recently, she has been examining the molecular mechanism of action of carotenoids, both vitamin A and non-vitamin A against several chronic diseases. Dr. Len, over to you.
All right. Thank you very much, Haneen. Well, it's my pleasure to introduce Dr. Paige Berger, who has a PhD and is also a registered dietitian. She's an investigator of pediatrics at Brigham and Women's Hospital and an instructor of pediatrics at Harvard Medical School. Dr. Berger's research examines the roles of infant nutrition and human milk composition on brain growth and maturation via magnetic resonance imaging. Her current work investigates the impact of human milk oligosaccharides on structural brain development in preterm infants, who are uniquely vulnerable to brain injury and neurodevelopmental defects. Yes, over to you, Paige.
Thank you so much, Dr. Asman. I really appreciate that introduction. It's a pleasure to be here this afternoon, I know in Jordan, and I just want to thank the Jordan Nutrition Innovation Lab for the opportunity to present my work. So as Dr. Asman mentioned, I'm going to be talking today about the state of the science on carbohydrates in human milk and the developing infant brain. Briefly, these are my disclosures. And these are my objectives. So today we're going to be talking about the influence of human milk carbohydrates on structural and functional brain development in human milk-fed infants. And at the end of this presentation, I hope that you will meet the following learning objectives. The first is to understand that human milk is a complex biological system that contains many, many components which may interact to affect health trajectories. The second is to recognize that carbohydrates are the most abundant solid constituent of human milk, and they have garnered attention in relation to early health outcomes. Also, it's to understand the role of maternal diet, maternal genetics, just these characteristics in general in determining the carbohydrate composition of human milk, and then be able to describe the state of the science on human milk carbohydrate exposure and the developing infant brain. So let's start with human milk in general. Human milk is the recommended diet for virtually all infants. And this recommendation is based on extensive evidence that breastfeeding and human milk exposure in general will reduce the risk for morbidity and mortality in the first few days and months of life. But in addition, studies have shown that human milk exposure early on may be also linked to additional health benefits. So studies support the role of human milk exposure on brain maturation and better neurodevelopmental outcomes, on immune system maturation and reduced risk for infections, physical growth and reduced risk for obesity, metabolic health and reduced risk for diabetes, and self-regulation of intake, and then better appetite control. Now, despite all of these purported benefits of human milk exposure on early growth and development, there are major knowledge gaps regarding the specific human milk components that may contribute to favorable outcomes or may also dampen the benefits of human milk exposure, which we'll discuss today. And these knowledge gaps really need to be addressed if we're going to maximize the therapeutic potential of human milk and early life nutrition in general. So first, I want to talk about the state of the science on human milk exposure and neurodevelopmental outcomes before we launch into a discussion of the specific components of human milk. So studies have shown that a longer duration of breastfeeding is associated with improved cognitive development at school age and beyond. So for example, studies have shown that breastfeeding for at least 3 months is associated with better problem solving and communication during later infancy. And studies have also shown that a greater duration of breastfeeding during infancy is associated with things like a higher IQ, better educational attainment, higher income level at 30 years of age as well. So these are evidence of long persistent benefits of breastfeeding early on. Now, the extent to which breastfeeding and human milk exposure confers these benefits to the brain is still a topic of discussion. And this is because the strength of the relationships between human milk exposure and our cognitive outcomes have been shown to vary from study to study. And these differences in the magnitude of these relationships may be attributed in part to biological and environmental influences of human milk composition, which are still not well defined and may increase or decrease the neuroactive potential of human milk. And so perhaps due to the dynamic nature of human milk composition and the diverse factors that shape human milk profile, the compounds in human milk that may be most beneficial for the brain are also poorly characterized and not fully elucidated. And so this knowledge gap needs to be addressed if we're going to, again, optimize the therapeutic potential of human milk exposure and ensure optimal brain structure and function in human milk-fed infants. Investigators in the field of human milk biology have taken steps toward understanding the biological and environmental factors that may shape human milk composition. And these steps require us to shift our research focus toward appreciating that human milk composition doesn't stand alone. It is part of a co-adapting system that we refer to as the mother-milk-infant triad. So specifically, maternal physiology, human milk composition, and infant physiology are all related, and they may ultimately affect the trajectory of maternal and infant health. And using this more expansive and comprehensive approach may lead to renewed efforts and successes in maximizing the therapeutic potential of human milk and optimizing early infant outcomes. So in our discussion of human milk composition today and the developing infant brain, I'm going to be highlighting maternal diet and genetic factors that may ultimately modify the composition of human milk and specifically the carbohydrate composition. And understanding the role of maternal physiology on human milk composition is really important because it may help to guide the development of study design and methodological considerations in research. And it may also help lead to more novel findings that provide new insights into factors that shape human milk composition. So of the many, many components in human milk, carbohydrates, again, are the most abundant solid constituent. And the major carbohydrate in human milk is lactose. Lactose is a disaccharide that is made up of two monosaccharides, which are glucose and galactose. And notably, glucose is the main source of energy for early brain development. But the carbohydrates in human milk have been shown to extend far beyond just lactose. And recent studies have demonstrated this. So, for example, fructose is a structural isomer of glucose, and our research team has shown that fructose is actually detectable in human milk. Now, this is interesting because fructose is not naturally produced by the body, but it is a component of whole fruits and high fructose corn syrup, which is a sweetener that is prevalent in sugar-sweetened beverages. So today I'm going to be discussing findings related to the inadvertent transmission of fructose from the maternal diet to maternal milk and the potential implications for exposure among human milk-fed infants. I would like to note, though, that not all carbohydrates are created equal, and different carbohydrate structures may have different physiological functions related to early brain development. So specifically, human milk oligosaccharides, or HMOs, are complex carbohydrates with over 200 distinct structural permutations. Unlike fructose, HMOs are naturally occurring in human milk, and they have been implicated in a wide range of beneficial health outcomes in human milk-fed infants. So the next few slides, we're going to talk about early exposure to fructose and its associations with brain development in human milk-fed infants. And then I will provide an overview of HMOs and their purported roles in infant brain structure and function. So fructose is a staple of the Western style diet, and I know that it is also highly prevalent in Jordan. Fructose is the main component of high fructose corn syrup, which is the leading source of added sugar in processed foods and also in soft drinks. Now, given its pervasiveness in our food supply, we conducted a randomized controlled trial of breastfeeding women to determine the effects of a high fructose corn syrup sweetened beverage on the carbohydrate composition of human milk. And I present these results in the figure to the right. We have human milk concentrations of fructose on the y-axis and time in minutes on the x-axis. And what we found was that breastfeeding women who consumed the high fructose corn syrup sweetened beverage had increased human milk concentrations of fructose within 2 hours after drinking their beverage. And these human milk concentrations of fructose remained elevated above baseline for up to 5 hours after intake. Now, these findings raise some potential concerns. And this is because animal studies have shown that rat pups exposed to high fructose corn syrup exhibit neuroinflammation in the centers of the brain that foster learning and memory. So it's possible that inadvertent transmission of fructose from the maternal diet to maternal milk may have unintended consequences on early brain development in nursing infants. So we decided to conduct a study to determine the associations of maternal fructose intake during this period of breastfeeding or lactation with cognitive development scores in their human milk-fed full-term infants. We also aimed to determine whether fructose from sugar-sweetened beverages and juices was related to our neurodevelopmental outcomes, or was it fructose derived from whole fruits? Because we know that traditionally whole fruits are considered a potentially healthy item in our diet, whereas sugar-sweetened beverages, not as healthy. They both contain fructose, so which may be affecting the brain. Our participants were mothers and their human milk-fed infants living in Los Angeles, California. And our exposure variables were collected at 1 and 6 months using a 24-hour dietary recall. We were specifically interested in maternal intake of fructose, sugar-sweetened beverages and juices, and whole fruits at 1 and 6 months. And this period is the recommended period of exclusive breastfeeding. Our outcome variables were collected at 24 months using the Bayley scales of infant and child development. And we were specifically interested in the cognitive domain of the Bayley scales, which assessed sensory motor integration, concept formation, attention, habituation, and memory. And the top figure to the right shows the timing of data collection. The bottom figure shows examples of cognitive assessments, which included tasks that engaged the infant in shapes and puzzles and things like that. And these are our results. So the table shows our findings using linear regression analyses. And we reported unstandardized beta coefficients, 95% confidence intervals, and p-values. And I've highlighted models 1 and 2 in this red box. And the data show that maternal intake of fructose at 1 month was significantly and inversely associated with infant cognitive development scores at 24 months. Here we have model 3 highlighted in red. And now these data show that maternal intake of fructose was no longer associated with infant cognitive development scores once sugar-sweetened beverages and juices was added to the model. Maternal intake of sugar-sweetened beverages and juices, however, was still significantly and inversely associated with infant cognitive development scores. And in addition to these findings, we also observed that maternal intake of whole fruits at 1 month was not significantly associated with infant cognitive development scores at 24 months. So overall, we found that greater maternal intake of fructose during early lactation was associated with poorer cognitive development scores in human milk-fed infants. And this inverse association was attributed to maternal intake of sugar-sweetened beverages and juices rather than whole fruits, which again are the leading sources of fructose in the diet. So, given our findings with transmission of fructose to human milk from the maternal diet, and given these findings showing that moms who eat more fructose and drink more sugar-sweetened beverages have children with lower cognitive development scores, that's the story that we were able to kind of uncover in this line of investigation. Now, there is a caveat. So in the previous study, we speculated that maternal intake of fructose from sugar-sweetened beverages during lactation may contribute to poorer cognitive development in their human milk-fed infants. But there is an alternate hypothesis, and that is that maternal fructose and added sugar consumption during lactation may actually reflect enduring dietary patterns that had persisted from pregnancy. So we conducted a study to determine associations between maternal added sugar intake during pregnancy and brain MRI outcomes in newborn infants. And some of our results are presented in the figure to the right. So these are statistical maps of the infant brain at 3 weeks of age. And these statistical maps show that maternal intake of added sugar during the second trimester of pregnancy was inversely associated with MRI measures of mean diffusivity in areas of cortical gray matter in the brain. I'm going to discuss the biological interpretation of our MRI-based findings in future slides, but for now, I will say that these results indicate that in utero exposure to added sugar may reduce neurobiological processes that form the structural basis for future learning and memory. And these data are in line with findings from our prior study, which show that maternal intake of fructose and sources of added sugar may adversely influence neurodevelopmental outcomes in their infants. So now I'd like to shift gears to a different group of human milk carbohydrates that may be more beneficial for the developing infant brain. HMOs, human milk oligosaccharides, are a group of unconjugated and undigestible glycans, or complex carbohydrates. And as I mentioned, over 200 distinct HMO structures have been identified to date. Now, what might come as a surprise to some of you is that HMOs are the third most abundant solid constituent of human milk after lactose and lipids. And if you combine HMOs and lactose, you see that carbohydrates are the most abundant constituent of human milk. HMO biosynthesis is also very unique, and this may be particularly important for brain development. So HMO biosynthesis begins with a basic structural blueprint. All HMOs contain lactose, which can be elongated. This process is depicted in the figure to the right, figure A, on the top. The lactose molecule can also be fucosylated, sialylated, or acetylated, and then these slight structural differences lead to the various HMO subgroups. So I've shown some of the examples of HMO structures in figure B. The top row shows 2 fucosylactose and 3 fucosylactose, which are fucosylated HMOs and their structural isomers. So they're identical except for the location of their fucose residues. We also have in the bottom row 3 sialactose and 6 sialactose, which are also structural isomers and sialylated HMOs, and they only differ in the location of their sialic acid residue. So notably, these slight structural differences may actually dictate diverse physiological functions, which may be especially important for brain structure and function. So as I mentioned earlier, it's important to understand the biological and environmental influences of HMO composition. And the reason for this being that these factors may inform the timing of human milk collection and analysis and other components of study design and methodology when we're planning this research. Studies have shown that the major determinant of HMO composition is maternal genetics, and HMO profile may mirror blood group characteristics. So for example, women who have an active secretor locus that encodes for a functional FUT2 enzyme, these women are classified as secretors. And secretors produce milk that is abundant in alpha-1,2 fucosylated HMOs, which include 2FL, like I discussed on the previous slide, as well as LNFP1. And those who lack a functional FUT2 enzyme are classified as non-secretors, and non-secretors produce milk that does not contain alpha-1,2 fucosylated HMOs. In addition to these FUT2 dependent HMOs, most other HMO concentrations also differ between secretors and non-secretors. So you're dealing with 2 essentially different HMO profiles of milk. In the table to the right, I've highlighted differences in HMO concentrations between secretor and non-secretor groups. And if you can see the 2 HMOs that are first highlighted, 2FL and LNFP1, they are significantly higher in secretors relative to non-secretors. And again, this is because they are FUT2 dependent HMOs. I've also highlighted here the HMO DSLNT because DSLNT concentrations do not significantly differ based on secretor status. And this is because DSLNT does not contain any alpha-1,2 linkages, so it's not a food-2 dependent HMO. Now, it's well established that HMO composition is affected by secretor status, but it's unclear whether infants who are born to secretors versus non-secretors and therefore are exposed to a different HMO profile also would differ in their growth and developmental outcomes. We're not there yet. So now let's move on to more of the topic at hand, which is how are HMOs associated with the developing infant brain? The potential mechanism is related to the way in which HMOs are handled in the gastrointestinal tract. HMOs are ingested during human milk feeding, and they reach the distal small intestine and the colon relatively intact. They are then selectively fed on by beneficial bacteria that populate the intestinal microbiome. And for those of you who might be unfamiliar, the gut microbiome refers to communities of microorganisms that may be helpful or harmful to host health trajectories. These microorganisms affect host physiology, including nutrient absorption, energy extraction, and immune regulation. To date, experimental studies have really focused on specific HMO structures, which I presented previously, that include 2FL, 3FL, 3SL, and 6SL in relation to brain development. So for example, studies have been conducted in rat pups and piglets, and they found that exposure to these specific HMOs led to improvements in learning and memory on certain tasks that the animals were put through. These findings were attributed in part to the role of HMOs modulating the gut microbiome in these animals. In addition, in vitro studies have shown that intestinal cells treated with 2FL, 3FL, 3SL, and 6SL had increased growth of beneficial bacteria, which included Lactobacillus, Bifidobacterium, and Bacteroides species. And these beneficial bacteria have been shown to produce neuroactive metabolites, including neurotransmitters and short-chain fatty acids, that may have far-reaching effects on the brain. So, for example, these neuroactive metabolites affect the enteric nervous system in the gastrointestinal tract, and they may also be absorbed by colonocytes and enter vagal pathways to the brain. So ultimately, these would affect neurobiological processes in brain development that may include regulation of brain-derived neurotrophic factor, or BDNF, which supports the differentiation of neurons or brain cells, and also synaptic plasticity and circuit formation that is integral to later learning and memory. And also the induction of T regulatory cells, interleukins, and other immune factors, which may enter systemic circulation and ultimately reduce neuroinflammation, which would be protective of brain development. This is an illustration. I like a visual. So this is an illustration of the potential mechanism through which HMO exposure may affect brain structure and function. You can see we have our four main HMO structures at the tippy top. These arrive in the distal small intestine and the colon relatively intact, increasing the growth and proliferation of these bacteria. Bacterial fermentation of HMOs leads to the production of neuroactive metabolites. These enter vagal pathways to the brain and affect neurobiological processes. And these neurobiological processes ultimately form the basis for future learning and memory in toddlerhood, school age, and beyond. So last year, we published a scoping review on associations of HMO exposures with neurodevelopmental outcomes in human milk-fed infants. And through this process, we identified 6 studies, including one of our own papers that was published in PLOS One, which were all observational in design and predominantly conducted in human milk-fed full-term infants. Exposure variables in these studies were defined as total and individual concentrations of HMOs, or relative abundances of HMOs, and they were measured at 1 and 6 months, which is this period of recommended period of exclusive milk feeding. Outcome variables were defined by several domains of neurodevelopment, which included cognitive, language, and motor skill development scores, and these were measured over 24 months of lactation. So overall, our narrative review found that greater exposure to total and individual fucosylated and sialylated HMOs, which included 2FL and 3SL, were associated with higher cognitive, language, and motor skill development scores between the ages of 18 and 24 months in human milk-fed infants, suggesting that earlier exposure to HMOs may confer beneficial outcomes for brain function later in infancy. Now, based on these observations, we hypothesize that favorable influences of HMOs on cognitive function may actually be attributed to earlier influences of HMOs on structural brain development. So we conducted a study to determine associations of HMO exposures with MRI measures of structural brain development in human milk-fed infants. Participants were mothers and their human milk-fed full-term infants, again, living in Los Angeles, California, and our exposure variables were collected at 1 month. We analyzed human milk samples collected at 1 month for the 19 most abundant HMOs using high-performance liquid chromatography. And based on the literature, our HMOs of initial interest were 2FL, 3FL, 3SL, and 6SL, although I'm going to be focusing on findings related to 2FL and 3FL just to make sure we have enough time to get through a lot of this work. Our outcome variables were also collected at 1 month, so this was a truly cross-sectional study. We obtained brain images using a 3T MRI scanner in infants, and we extracted diffusion measures of brain tissue microstructure, including fractional anisotropy and mean diffusivity values. And I'm going to describe these outcome variables in greater detail in the coming slides. So we use several MRI modalities, but today I'm going to focus on our findings using diffusion tensor imaging, or DTI. So DTI measures the movement of water that is going to be modulated by characteristics of brain tissue microstructure. And DTI is indexed by FA and MD values. FA, or fractional anisotropy, is the movement of water in a single direction. And very simply put, in areas of developing white matter, higher FA values may reflect neurobiological processes of myelination. Myelin is the lipid sheath that surrounds the axon of a brain cell, and greater myelination is going to increase the speed of signals between brain cells or between neurons. In contrast, MD is the diffusion of water, or mean diffusivity is the diffusion of water in all directions without directional specificity. And in areas of cortical gray matter, higher MD values may reflect greater dendritic arborization and synaptogenesis, which are a different set of neurobiological processes important for brain development. Specifically, dendrites and the synapses that they support increase the connectivity between neurons and also underlie normal brain function. And so I have tried to illustrate the influence of neuron or brain cell structure on water diffusion in the figure to the right. As you can see in the body of the neuron, it contains dendrites that branch in all directions without directional specificity. And so greater dendritic arborization may be reflected in higher MD values, MD being the measure of water diffusion in all directions without directional specificity. You can also see that the axon of the neuron is coated in myelin, a lipid sheath that is hydrophobic. And because it is hydrophobic, myelin constrains the movement of water in a single direction that is parallel to the long axon of this neuron. So greater myelination, which is going to increase water diffusion in a single direction, may be reflected in higher FA values, which measures the movement of water in a single direction. Overall, what I just described, dendritic arborization, is the structural basis for learning and memory, and myelination is essential for information transfer and higher order cognitive functions. So this is a simple overview of the ways in which water diffusion is modulated by neuronal structure and then how neuronal structure relates to cognitive function. And these are our first set of results. I want to make sure I have time for everything, so I'm just going to go through these briefly because I think the biological interpretation is very important. But these are statistical maps depicting associations of 2FL concentrations with FA and MD values in the infant brain. Within each row, the first image represents the innermost slice of the infant brain, and the map all the way to the right is the outermost slice. Our positive associations between exposure and outcome are defined by warm colors, and our inverse associations are defined by cool colors. Only only p-values that survived FDR correction are plotted here. So overall, we found that 2FL concentrations at 1 month were significantly associated with FA and MD values throughout the cortical mantle of the brain. So here I highlight associations of 2FL concentrations with FA values. You can see there's a significant inverse association in areas that likely included the dorsolateral prefrontal cortex and the posterior cingulate cortex, so areas of cortical gray matter. Panel B shows a significant positive association of 2FL concentrations with MD values located in similar areas of posterior cortical gray matter, as well as in posterior white matter and subcortical gray matter nuclei. And the cortical gray matter areas of interest likely included, again, the posterior cingulate cortex, as well as the lenticular nucleus, which is a subcortical gray matter, which is in subcortical gray matter nuclei, so deeper in the brain. Collectively, these results suggest that 2FL concentrations are associated with lower FA values and higher MD values in areas of cortical gray matter. And these are statistical maps depicting associations of 3FL concentrations with FA and MD values in the infant brain. And overall, we found that 3FL concentrations were significantly associated with FA and MD values, but this time in areas of developing white matter. So panel A shows a significant positive association of 3FL concentrations with FA values in the anterior corona radiata, the internal capsule, and the corpus callosum. Again, these being largely white matter tracts in the infant brain. Panel B shows a significant inverse association of 3FL concentrations with MD values located in similar regions of developing white matter, again including the anterior corona radiata, the internal capsule, and the posterior corona radiata. So collectively, these results show that higher 3FL concentrations are associated with higher FA values and lower MD values in areas of developing white matter. So now that we've reviewed the findings, I'll talk a little bit about the biological significance of our results. So overall, we found that slight structural differences in HMOs may confer distinct but complementary influences on brain development. So we found that higher concentrations of 2FL were associated with lower FA values and higher MD values in areas of cortical gray matter. And these findings suggest that greater exposure to 2FL may be associated with greater dendritic arborization and synaptogenesis. Again, you can see dendrites are branching in all directions without directional specificity. And so it's plausible that greater dendritic arborization would increase the diffusion of water in all directions without directional specificity, as we observed in relation to 2FL concentrations. Again, dendritic arborization is the structural basis for future learning and memory. So that is why it's important for cognition. We also found that higher concentrations of 3FL, the structural isomer of 2FL, were associated with higher FA and lower MD in areas of developing white matter this time, which suggests that 3FL may be associated with more rapid myelination in the infant brain. You can see that myelinated axon is coated in lipid, and that lipid sheath is hydrophobic. And so myelin constrains the diffusion of water in a single direction parallel to that long axon. So it's plausible that greater myelination would increase the diffusion of water in a single direction, as we observed in relation to 3FL concentrations. And so it is important to note that more rapid myelination is associated with more rapid information transfer in the brain, which is necessary for higher order cognitive functions. Again, I recap here that dendritic arborization is the structural basis for future learning and memory. Myelination is essential for higher order cognitive functions. So overall, our data show that individual HMO exposures defined by their distinct structural characteristics differentially associate with MRI measures of structural brain development in human milk-fed infants. And our findings specifically highlight roles of 2FL and 3FL in promoting the maturation of cortical gray matter and developing white matter. Now, I don't have time, unfortunately, to present this as our latest work. We've shifted gears now, and we're looking at HMO exposure in very preterm infants who are particularly vulnerable to brain injury and dysmaturation and may therefore benefit most from HMO exposure. We have been conducting studies to determine associations of HMO exposure during NICU hospitalization with our MRI outcomes, but this time of total and regional brain volumes or sizes. And again, this is our study design, but I'll skip to the end, which is that we ultimately so far have found that higher concentrations of 6SL, which is another HMO that I briefly mentioned, were associated with larger total brain volumes in very preterm infants at term-corrected age, suggesting that 6SL may hold promise for promoting brain growth during a critical period of brain development during hospitalization. So this is my overall summary. I'm at the end now. Overall, human milk is a complex biological system. It has both nutritive and non-nutritive bioactive effects on the brain. Carbohydrates are the major constituent of human milk and may affect diverse physiological functions in brain development. Fructose is derived from the maternal diet and may confer adverse influences on brain development, while HMOs hold promise for promoting brain growth and maturation. Thank you very much, and I'm going to stop sharing.
Okay, thank you very much, Dr. Berger. That was absolutely fascinating. Okay, now we have time for some questions. And I already see one question in the chat. Have you studied any non-breastfed, formula-fed infants who would get no HMOs?
That is a really good question. So right now, I think this work is focusing on human milk-fed infants with the purpose of seeing what components of human milk may confer benefits for brain development that we could then lead to maybe new nutritional products that would put all infants, whether they have access to breast milk or formula, on the same level playing field. Interestingly, formula companies have started to add one HMO, which is 2FL, to their products. But in order to add potentially more HMOs, we first need to do the research and come up with evidence-based guidance for those to kind of enter into the commercial space, essentially. So we have not focused on formula-fed infants at this stage, but I will say that I think we're working toward understanding HMOs more so that we can also help get formula-fed infants access and exposure as well.
All right. Okay, thank you. I think from Dr. Kelly Dingus, for the data you presented, is the secretor status determined by the milk HMO profile or by maternal genotyping? I am also interested what you think about the impact of your results on the infant secretor status. If a secretor mother-infant pair would have the best brain development outcomes, seeing the importance of myelination in relation to 2 and 3FL.
Another great question. So there are studies that are starting to genotype moms to really confirm their secretor status. In our work, and we collaborate with Dr. Lars Bode from the University of California, San Diego, who's a pioneer in the area of HMOs. He assesses secretor status by looking at those alpha-1,2 fucosylated HMOs in milk. So mothers who are secretors produce 2FL, for example, and LNFP1. Those that are non-secretors do not. So that is how we sort of assess maternal secretor status, is by what are the concentrations of those key HMOs in milk. you are absolutely right that infants are also secretors or non-secretors. And I think that understanding that match or mismatch between mother and infant secretor status would be very helpful and informative in terms of which is the best combination. But I don't think we're there yet, but we'll hopefully get there.
Okay, keep the questions coming in. The answer is, given what you just said, are diet recommendations for lactating mothers? Are we telling mothers not to have SSB or what?
So I come from the school of breastfeeding is a very, can be very challenging, and we don't want to perhaps deny mothers the opportunity to consume something that brings them happiness and joy and that they like to consume. But we do need to be mindful of the potential implications on the infant. And so perhaps future dietary guidance, if we do more research in this area and really solidify these findings, would recommend that maybe mothers be more cognizant of the timing of their SSB consumption relative to their feeding of the infant. Not to say that the implications of fructose exposure in milk are the same as, let's say, alcohol exposure in human milk, but we do have these guidelines about drinking alcoholic beverages surrounding breastfeeding infants. And so perhaps there's a day when we really, again, do more work in this area and see what the negative implications may be, that we also recommend that mothers be, again, consuming SSBs, but like being mindful of feeding their infant. And so maybe not doing that when they're about to breastfeed.
Okay, now, do we have another question? Not yet. Okay, keep them coming. Well, I have a lot actually, but in thinking about sugar sweetened beverages with high fructose corn syrup, what about eating cakes and pies, things that have sucrose, which is glucose and fructose? Do we have any information on like the regular diet? Forget about, the SSBs.
Yeah, I think that work is largely derived from animal studies. When animals are fed like a very sucrose concentrated, water, whatever it may be. And I think there is some evidence that is also potentially leads to adverse outcomes in terms of, brain structure and function. I'm not as familiar with that literature. The reason that we focus on SSBs is just because, at least in the US, it is our most prevalent source of added sugar. But I think that, and to your question, Dr. Ausman, about cakes and pies and things, I don't know if that would have the same effect on breast milk composition, only because that sugar is baked into so many other ingredients and so many other macronutrients that perhaps it slows down the digestion, and therefore it enters breast milk differently. I do want to note that the reason, though, we're interested in fructose is also because fructose is metabolized in the body differently than glucose. Which is that glucose is regulated by insulin, right?
Mm-hmm.
So that is going to help to maintain blood sugar concentrations, whereas fructose is not regulated by any hormone and it goes straight to the liver. So that's another reason we're interested in fructose, is there isn't anything in the body that's perhaps regulating fructose and fructose entering circulation and breast milk composition. And whereas glucose is regulated by insulin, perhaps before it enters breast milk.
Okay, the questions are piling up. Okay, I have another question. Can you speak to the 3SL data that you didn't present?
Yes, I can. Thank you for that question. So we did also find significant relationships between 3SL concentrations with our fractional anisotropy and mean diffusivity outcomes. And we saw these associations again in areas of developing white matter. So this would suggest that 3SL perhaps plays a similar role as 3FL in helping to promote greater white matter myelination, which again is important for higher order cognitive functions. So we did observe, 3SL was another HMO that perhaps holds promise for structural brain development.
Okay, thank you. Here's a clarifying question from one of our attendees. I thought fructose only reached the general circulation in hereditary fructose intolerance. How common is it to find fructose detectable in blood so it can reach the fetus or the breast milk?
That is again another good question. So fructose in and of itself can't be perhaps used for energy, and so it needs to be converted into glucose in order to be used for energy. And so, to answer that question, I'm not sure entirely of the physiology and the metabolic fate of fructose in terms of circulation and making its way to breast milk. All I can say is that we were able to assay our breast milk and found fructose concentrations in breast milk. Now, the concentration of fructose perhaps is not all that high, but a similar dose of fructose in our breast milk has been reported in animal studies to increase things like adipogenesis and have other physiological roles in the body, which suggests that it could still be relevant. But in terms of the actual metabolism, that is a little bit out of the scope of my work.
All right, thank you. Now here's a very interesting question. Thank you for presenting this important topic. Are there any studies that compare the effect of oligosaccharides in camel milk with those in human breast milk on cognitive development?
Interesting question. I know that there are studies that look at oligosaccharide profile in animals relative, and what they found is that things like bovine oligosaccharides, they're different than human milk oligosaccharides. I can't speak to camels, but I think that generally the oligosaccharides that are produced in animal milk relative to human milk are not exactly the same. Now, do they still have potential implications for growth and development of the animal? Perhaps. But I don't know. It's difficult to compare species and HMO profiles in that sense and their implications in terms of health outcomes.
Okay, one of the attendees would like the links to the studies. I think you're going to make available to Maha the reference list, right?
Yes.
So that people can do that. Okay.
Yes. So I will put that at the end of the slide deck and send
All
it.
right. So what's to stop a company from making a bunch of these oligos and putting in them form of pills? And then mothers who are breastfeeding who are non-secretors can have some of these things. Has anyone thought about that?
Yes, I think part of the trouble right now is that 2FL, which is being added to Similac and other types of commercial infant formulas, is relatively inexpensive and straightforward to manufacture. And I think that trying to manufacture some of these other HMOs has been a bit more challenging. And so that is right now a current gap that needs to be addressed. And, in terms of would a mom who consumes an HMO tablet, would that ultimately arrive in breast milk and make itself available to the infant? I don't know if we know the physiological fate of those HMOs and whether it would have the same, effect on breast milk. And also, it's important to note that we know that genetics determine HMO profile in human milk, but we don't actually know what HMO profile is the best and would produce the most robust influences on infant brain structure and cognitive function. It's also possible that as somebody alluded to with the maternal secretor and infant secretor status, that milk is made specifically for that infant for reasons that are perhaps outside of just brain development. And so there's a biological, purpose and importance of that milk specifically for that child. But again, we don't know yet.
All right, thank you. Have you looked at the non-secretory HMOs in relation to infant brain development?
That's a really good question. So I would say that the 2FL and the 3FL are fucosylated HMOs, and so they're produced mostly by secretors. 3SL, I think, is also different between secretors and non-secretors, but I don't believe that 6SL is. And as you saw, we did not see an association, or you didn't see, but I will tell you that we did not actually observe an association between 6SL concentrations with brain MRI outcomes in full-term infants. But as I just showed, we did observe a significant association of 6SL concentrations with brain development outcomes in preterm infants. And so we have examined, some non-FUT2 dependent HMOs. And I think that we have a lot of data, and that is a next step for us, certainly. So please stay tuned.
Okay. Now, have you looked at any in utero work and using the techniques you've described?
So I personally have not. I do believe that there is HMO exposure, specifically in utero, through amniotic fluid. But again, I'm not as brushed up on that area of research. Also, we can't do a lot of the brain imaging techniques in utero. We can do things like ultrasound or whatnot, but again, a little bit out of the scope of my work. And then in terms of, exposure to things like added sugars and fructose, we have done some of that work to look at maternal diet during pregnancy and brain outcomes in newborns. And we have seen that there is an association between maternal intake of added sugar in the prenatal period with brain outcomes in the postnatal period of infants.
Okay. Oh, that's fascinating. All right. I have one more question. Some of your outcomes are at 1 month and some were at 6 months. Is there a difference if somebody breastfeeds for just 3 months and then goes on to are there differences that you can detect if people stop breastfeeding before 6 months?
That's a good question. So we focused on exposures at 1 month and 6 months because that's the recommended period of exclusive breastfeeding. A lot of our findings showed that earlier exposure at 1 month were associated with our outcomes, but not at 6 months. And so the idea is that either it's the timing of exposure really early on in the infant's, postnatal life that's important for influencing their brain development, let's say, versus the later exposure at 6 months. Or it could be that as, moms initiate breastfeeding, they might be breastfeeding more earlier on, and then breastfeeding may taper with the introduction of solid foods. And so infants are not getting exposed to the same components of human milk that they did earlier on. So our work does suggest that there is a temporal window of, let's say, HMO exposure that is particularly beneficial for brain development. And similar with the with fructose, that there is a temporal window when it may be more be potentially more leading to adverse outcomes the earlier they're exposed.
All right. Well, I think it's time for summary, and I just realized you're the best person to give us like a few sentences, take-home messages in our remaining couple of minutes.
I would be happy to. So overall, I would like to say that human milk feeding, breastfeeding is going to be the best source of nutrition for infants through the first 6 months of life. But that breast milk, that human milk, isn't, so easy to kind of understand. It is a biological system. It does have many, many components that may be contributing to these health outcomes. I've identified just a few components that all are based on carbohydrate structure that have been associated with brain structure and function. And, it's likely that maternal characteristics and maternal influences may modify milk carbohydrate composition to make it more beneficial for the brain, or maybe dampen its effects on the brain. And so we should be cognizant of those factors, but continue to breastfeed our babies.
Well, thank you very much. This is absolutely fascinating. More questions have come in, but I think we can send them by email. And I want to thank everyone else for attending. Any final words from Haneen or anyone else? Okay, thank you very much, Dr. Berger. This
Thank you.
was great. Oh, by the way, there is the camel milk O's have been done, and we can send you what they have found.
Thank you so much. I appreciate that.
Yeah. All right, thank you. Bye-bye.
Thank you. Bye-bye.